In Drosophila oocytes, several mRNAs are specifically sorted to distinct parts of the cell, where they will establish the future body axes of the developing embryo after fertilization. The mRNA oskar, for example, is transported by the +N-directed microtubule motor kinesin 1 to the oocyte posterior, where it will specify the embryonic posterior and germline. Ilan from the University of Oxford in the UK has long been interested in how mRNAs like oskar get to their specific locations.
The underlying organization and polarity of the microtubules is really essential for dictating where cargoes end up. Despite this, and despite a lot of work by many investigators, we didn't know precisely the global organization and polarity of microtubules, to what extent the microtubules were dynamic, and to what extent they have a stereotypic distribution of polarities from anterior to posterior. So we decided to tackle this and actually determine the global distribution and polarity of microtubules.
The project was led by postdoc Richard Parton.
We understood very quickly that it was very important to image the organization in live tissues. This overcomes problems with poor preservation of the microtubule network you often get with fixation. And so to work with these live cells required us to carefully optimize the preparation of the tissue to maintain viability, and also fine-tuning of the imaging system to improve the imaging quality.
Parton began by imaging oocyte microtubules labelled along their entire length with the microtubule binding protein tau-GFP.
One of the first things we noticed was just how complicated and dynamic this organization was. And we also noticed how these microtubules extended right through the oocytes into the posterior regions.
Microtubules weren't as abundant in the oocyte posterior, but microtubules throughout the cell were highly dynamic, as visualized here by the +N tracking protein EB1.
We wanted to convince ourselves that these dynamic microtubules were actually relevant in localizing cargoes, and what we did, we performed extended simultaneous time-lapse imaging of both the microtubule and moving cargoes.
Specifically, Parton and colleagues labelled microtubules and the RNA binding protein staufin, which is essential for transporting oskar mRNA to the oocyte posterior. The researchers saw staufin particles moving along unstable dynamic microtubule filaments. But how are these filaments organized so that kinesin 1 transports staufin and oskar to the oocyte posterior? Parton and colleagues examined the microtubule's orientation by tracking the movements of the +N marker EB1.
So when I first started analyzing the EB1 data, I was doing it by hand, and it very quickly became apparent that there's no way we could get an adequate amount of quantitative data simply doing it manually. It was taking up to 20 hours to analyze one data set.
So the study involved a number of computational people to develop new tools to visualize and statistically analyze the polarity of microtubules. So Graham Ball and Russell Hamilton were the two people in our lab who worked on this. Graham developed methods for picking out the moving particles out of a background of a lot of fluorescence. And Russell developed tools to display and characterize a field of microtubule tips moving and give them an overall polarity in a sort of grid, which are referred to as a wind map, by analogy to a weather map where you can see the directionality of the wind.
This approach, also developed in collaboration with Wai-Ping Liu and Lei Yang at Heriot-Watt University in Edinburgh, allowed the researchers to determine that the seemingly random and chaotic network of oocyte microtubules was actually subtly biased so that around 60% of microtubule +Ns pointed toward the cell posterior. Moreover, this bias increased in regions of the oocyte closer to the cell rear.
So at the anterior, the polarities are much more random, whereas right at the posterior tip, they tend to be more polarized towards the posterior.
This microtubule arrangement, which the researchers call a biased random organization, is mirrored by the way in which oskar moves to the oocyte posterior, as originally shown by Daniel St. Johnston and colleagues, including Ilan in 2008. Oskar and staufin move randomly, but with a subtle bias towards the posterior, a bias that Parton et al. Now found became more pronounced in regions near the oocyte rear, just like the bias in microtubule polarity.
So we really wanted to understand how such a biased random organization was established and maintained.
Parton and colleagues investigated where in the oocyte microtubules are formed both by staining for the nucleating factor gamma-tubulin and by depolymerizing microtubules with the drug colcimid and then watching them regrow.
So using this technique, what we actually found is that microtubules are initiated from discrete foci that are around the cortex of oocyte, but are particularly dense at the anterior, and they decrease in density further from the anterior and are absent from the extreme posterior cortex. These foci radiate out microtubules in different directions and are able to extend into the posterior, but because of their absence from the extreme posterior, there are no microtubules extending back from the posterior cortex.
This decrease in microtubule nucleation from the anterior to posterior cortex of the oocyte sets up the biased random organization of the cytoskeleton. But how is this gradient of microtubule nucleation regulated? Parton et al. Turned their attention to the polarity protein PAR1.
It was already known that in PAR1 mutants, oskar mRNA was mislocalized, and that there was an effect on microtubule distribution. So that was a sort of hint. And since we now had tools which were much more subtle and much more discriminating in polarity, we could characterize the mutants much further in terms of the effect on microtubules.
In oocytes lacking PAR1, microtubule nucleation occurred all over the cell cortex, including at the posterior, and there was no longer a bias in microtubule orientation. Microtubules pointed equally toward both ends of the cell, potentially explaining why oskar mRNA isn't delivered to the posterior of PAR1 mutant oocytes.
It suggested that the bias random distribution was essential for distributing cargo to the posterior, but it also suggested that PAR1 was in some way essential for preventing nucleation of microtubules at the posterior. Now, we currently don't understand the mechanism by which that occurs. This is clearly a very important question that requires a lot more work.
But why organize oocyte microtubules like this? Why not maintain a strictly polarized cytoskeleton of stable microtubules that head directly from the anterior to the posterior of the cell? Davis and Parton think that the biased random distribution of microtubules might help oocytes to segregate components such as oskar to specific compartments of the cell, while distributing other components, such as organelles or generic mRNAs, evenly throughout the cytoplasm.
So we think that this microtubule organization provides a compromise between the need to mix everything together and distribute individual components to particular sites. If you had superpolarized highways of microtubules, then you'd end up with everything segregated. So I think in general there's been a slightly naive view of cells, and in general people think of cells as highly polarized, and perhaps using these new tools we've developed, we might discover in fact that in other cell types, in many cases, there's a subtle biased polarity rather than such a stereotypic clear-cut polarity.
The researchers also want to use their new analysis tools to investigate how certain mRNAs are transported to the oocyte anterior. But in the meantime, you can learn more about how a PAR1-mediated bias in microtubule organization directs mRNAs to the posterior of fly oocytes in the paper by Parton et al., published in the July 11, 2011 issue of the Journal of Cell Biology.